Hello students. In this video, we will learn about how our excretory systems works to create urine in our body. Let's learn. Now let's discuss urine formation, which happens in three main steps. The first is glomerular filtration, where blood pressure forces plasma through the glomerular filtration membrane, allowing water and small solutes to enter Bowman's capsule. The second step is tubular reabsorption, where essential substances like glucose, amino acids, and electrolytes are returned to the bloodstream from the tubular fluid. Finally, tubular secretion occurs, where additional wastes move from peritubular capillaries into tubular fluid and helps eliminate toxins and regulates blood pH. Step 1. Glomerular filtration. On an average, 1,100 to 1,200 milliliters of blood is filtered by the kidneys per minute, which constitute roughly 1/5 of the blood pumped out by each ventricle of the heart in a minute. The glomerular capillary blood pressure causes filtration of blood through three layers, which are the endothelium of glomerular blood vessels, the epithelium of Bowman's capsule, and a basement membrane between these two layers. Podocytes in Bowman's capsule are intricately arranged, forming tiny filtration slits. These allow all plasma components except proteins to pass into the capsule, making it an ultrafiltration process. In the regulation of glomerular filtration, healthy individuals have a glomerular filtration rate, GFR, of approximately 125 milliliters per minute, totaling 180 liters daily. The Juxtaglomerular Apparatus is a special area where the distal convoluted tubule meets the afferent arteriole. It senses blood flow and pressure, detects changes in GFR, and triggers responses to maintain balance. Next, we have tubular reabsorption. After glomerular filtration, the filtrate passes through the renal tubule, where reabsorption and secretion processes occur. These are crucial for maintaining the body's fluid and electrolyte balance. Nearly 99% of the filtrate is reabsorbed back into the bloodstream. Tubular epithelial cells in different segments of the nephron perform this function either by active or passive mechanisms. Then, glucose, amino acids, and sodium are reabsorbed actively. Nitrogenous wastes are absorbed by passive transport, and water reabsorption occurs passively in initial segments. Now let's discuss tubular secretion. During urine formation, tubular cells secrete substances like H+, K+, and ammonia into the filtrate. This process is vital for maintaining the ionic and acid-base balance of body fluids. Each segment of the renal tubule has specialized functions in processing the glomerular filtrate. The proximal convoluted tubule, PCT, is lined by simple cuboidal brush border epithelium that increases surface area. It reabsorbs nearly all essential nutrients and 70 to 80% of electrolytes and water, maintains pH by secreting H ions and ammonia, while absorbing bicarbonate ions. Henle's loop, minimal reabsorption in ascending limb, but crucial for maintaining high osmolarity of medullary fluid. Descending limb is permeable to water but not electrolytes, while ascending limb allows electrolyte transport but is impermeable to water. Distal convoluted tubule, DCT, performs conditional reabsorption of sodium and water. It also reabsorbs bicarbonate and selectively secreting hydrogen, potassium, and ammonia to maintain pH and sodium-potassium balance in blood. Now, let's talk about the collecting duct. This long duct extends from the cortex of the kidney to the inner parts of the medulla. Large amounts of water could be reabsorbed from this region to produce concentrated urine. It allows passage of small amounts of urea into the medullary interstitium to keep up the osmolarity and also plays a role in the maintenance of pH and ionic balance of blood by the selective secretion of hydrogen and potassium ions. Finally, we'll discuss the mechanism of concentration of the filtrate. The filtrate enters the descending limb and exits the ascending limb. Mammals have the ability to produce concentrated urine due to the counter-current mechanism provided by Henle's loop and vasa recta. The flow of filtrate and blood in opposite directions creates a counter-current pattern. The proximity between Henle's loop and vasa recta, along with the counter-current mechanism, maintains increasing osmolarity. The osmolarity gradient from cortex to inner medulla, ranging from 300 milliosmol per liter to 1200 milliosmol per liter, is primarily due to NaCl and urea. Let's take a deep dive into the countercurrent mechanism. NaCl is transported by the ascending limb of Henle's loop, which is exchanged with the descending limb of vasa recta. Then, it's returned to the interstitium by the ascending portion of vasa recta. Similarly, small amounts of urea enter the thin segment of the ascending limb of Henle's loop, which is transported back to the interstitium by the collecting tubule. In the medullary interstitium, presence of such interstitial gradient helps in an easy passage of water from the collecting tubule, thereby concentrating the filtrate urine. Human kidneys can produce urine nearly 4 times concentrated than the initial filtrate formed. We hope that you have understood the topic. You can now study chapter notes, watch video lectures, and solve MCQ tests of this chapter on EduRev to ensure you score well in your exams. And that's not all. You also get amazing courses for maths, science, English, Hindi, and much more for NEET. Thank you.